we have in the
Yes.
yesterday
Yeah, ma'am, you are audible.
yeah, in the yesterday class we understood that how genotyping is performed, how phenotyping is done, and what type, the details about the variant, what types of variant are. So today we will be starting with the hands-on session. Yeah, all of you have a Linux operating system, either Ubuntu or something else. No, all these runs of the today's session in which we have to identify variants from a sequencing data, that is from the genotyping data which was run on a sequencer. The output of that we have to process that. So you need a it will not work on Windows, it will only work on a Linux system. So I have given the links of the above softwares which I'll be using. One by one I'll be explaining you each, and whoever has Ubuntu can easily install them. Other Windows it cannot be installed. FastQC can be installed, but other software cannot run on Windows. Okay, so you want the link again? Okay, I'll share you the link. It is in the chat only.
Ma'am,
It
you
should be
start. You
visible.
can start. In the meantime, I will share the
Okay,
links.
okay, sure, thank you. So first I'll share my screen. So you all can see my screen.
It's visible, ma'am.
Okay. So today our session is the variant identification or calling from a reference-based genome. So during this session, we as we discussed yesterday, we have run our genotyping sample on a sequencer. Now we have the output of the sequencer of the sequencing run, and the sequencing run output contains FASTQ or FASTA files. So the steps which we'll be following in our today's session is the quality check of those FASTA files, the trimming of any of the uh, of low quality read in those FASTA files, mapping of the high quality genome, mapping it those reads to the high quality genome, and then the evaluation of the alignment, then identification of variant as the time permits, and their statistics, what means how many variants we have identified. That is a statistical check of the variants. Okay, so first of all, see how is the data obtained from the sequencer after the genotype run, like if you have run GBS or we have done RADSAC, all will give a output of the FASTA or FASTQ files. All the FASTQ file have a header. This header contains the information of the location of the
But
flow
ma'am,
cell.
your slide are not moving. They are freezed.
It's not moving.
You are in your introduction slide, slide number 1.
It is freezed in that.
Yeah, slide number
Okay,
1. So
I will share again.
yeah.
Now is it other slide?
You can once move your slides.
I have moved my slide. Are you
Yeah,
able
yeah.
to
No,
see the
no.
change?
Yeah, now they are moving.
Okay, okay. So the raw reads have a header in which there is information about the location of the run of your sample on the flow cell, and the quality which is represented. This is the sequence ATGC, and the location and the quality of each base obtained is in the lowest, in the fourth line. This way we get several FASTA files after a run, and that is specific to the sample. Like every sample will have several reads in each FASTA file. This is one FASTA file and there are so many reads in this. And now the the software which is very commonly used for checking the quality of the FASTA files, because we have received the FASTA file output from the sequencer after our genotyping run. But now a question is whether its quality is good or not. So the very commonly used software is FastQC, which tells about the quality of the reads, and also tells different parameters of the raw reads which may lead to problem in the analysis, like low quality reads or adapter contamination. All these it checks. So that now I have sent you all the files which we have to use today. I feel all of you have received and downloaded it. It was a 6.5 GB data around.
So I have shared the tar file in the drive link and also mentioned on the WhatsApp group.
Yeah. So now I will be first starting with this FastQC software. So if anyone has installed it, then it's good. Or if they have not installed, then I'm showing you how to install it. This is your Linux screen. Okay. So now if you want to install, if you have not installed, then you can use sudo apt. This will not work for all the system. I will give a common way to run for all of you. You are
But
able to,
your,
yeah,
your terminal is not visible, ma'am.
not visible.
Yeah. So what you
Uh,
can do, you can share your entire screen instead of
okay,
window.
okay. Optimize for video clip. Actually, I will share again and again because this is a Linux OS. So this doesn't give, it is not Windows. So it is not giving the option to share the entire screen at a time.
Okay, okay, ma'am.
Okay.
So
So
you
I
have to
will keep on.
switch.
Yeah, I will switch. I will keep
Yeah.
switching. So see, you open the link which I have given for the FastQC software. Okay. On this link, you will get the option of downloading this software. Okay. This can be done also by those who have Windows in their system. Once you download this, then let me know so that I can proceed with the next steps. If who have Linux can use this version, and Windows also the same version will be installed. So once it is done, let me know so that I can explain you how to go to the next step. The other option for you is also there. If you have Ubuntu 6 above 12 Ubuntu version, then you can also use sudo apt install and write FastQC. That will also install. I'm writing in the chat window. I will share my screen again. See, in the new share, I'm showing that you can write this command in which you can type sudo apt install FastQC. This will install your FastQC software. Mine is already installed, so it is saying that I have the latest version. Are you able to understand what I'm conveying?
Ma'am, is it possible to zoom the font size of your terminal?
Yeah, sure. Yeah, I will zoom.
So it will be better for recording
Yeah, I will
purposes
zoom.
as well as for
Yeah,
demonstration.
I will zoom. Sorry. plus. Is it visible now?
Yes, ma'am. It's better now.
Okay. If you want more, then I can zoom.
No, no, ma'am. It's good. It's good now.
Okay. So have all participants able to install this on Ubuntu or any of the Linux operating system or Windows?
Ma'am, is it possible to copy your? Can you copy your commands in the chat box so that
Copy my
I will share?
command.
Yeah, I will share
Okay,
this.
I'll copy my
I will
command.
share the entire chat history with our participants.
Anji, what are
So,
you saying?
ma'am, I am saying that kindly, if possible, paste your commands in the chat box. I will share
Yeah,
this chat box
yeah,
with
on
the
the chat.
participants.
Yeah,
Okay, ma'am. Thank you for your kind.
yeah, sure, sure. So done. Yeah, Bharti has done. So we can move forward. If all have done, well, that's great. So the next step, I'll explain you how to run this. You get, you type fast QC. It will open a window. Is open. This is a terminal based run. If you want to run several files at a time, you can be selected from here also, and you can give path direct to the directory also here. But I will explain like this because Windows user can also easily understand. So now you will open the file where it is located. I feel I will share the screen again. New share. You can see. I feel all can see my window of fast QC. If
Yes,
there is
yes,
any
ma'am.
problem,
Yes,
yeah,
ma'am.
then please stop me.
Yes.
Now you will go to the path wherever you have saved the file which I have sent you by email. So you will go to those file. In that I have actually given all the raw and all the analyzed files, but you will first currently use the raw file here. So you will see there is file named sample1, R1 and R2. This is sample1, R1 and sample2, R2. First, I will show the quality check of these two. This is the read 1 and read 2 for one sample. Okay. You will select and open it. Once it gets open, it will take some time. It will give you the result. Yes, see. Now we got the result that how is the quality, what is the basic stats? First of all is what is the basic stat of our file. Okay. So basic stat is that we have the input total sequence present is in our file is 6,87,599. The length of every read is 151, or means approximate length. Majority reads are of this length, and the GC content is 46. This is the stats of our total reads, which is an optimum and a good stat for the for a Illumina, because it's a Illumina short read sequencer which gives read in this range mainly. So it is a okay. That's why a green tick is shown by FastQC. Then comes the quality of your sequence. If you see the quality is measured by a Q30. I feel all of you must be knowing Q30 is a Phred score, what we call. Y-axis shows the Phred score, and x-axis shows the position of every base in the read. So what you will see that that the Phred score above 30 is considered to be very good quality read. So you see here that how is Phred score calculated? It is based on probability. Above Q30 means there is in 1,000 base pairs there is a probability of getting one base pair wrong. So that's why it's a high quality read. the base pair calling means the base which has been called by the sequencer, its quality is very high. So it's, you can see the majority of the bases, because the length of our read is 1 to 150. So the majority of the bases uh, called by the sequencer have high quality, which is above Q30. Okay, so this is observed here, and towards the end you can see that a little bit a dip in the quality is there, but it doesn't go below Q30, or rest are above Q32 also, but it doesn't go below. So overall quality of the forward read is good. Then if we see the sequence per style sequence quality, actually this shows the tile based sequence quality, which is the based on the flow cell location of that raw fast Q reads on the flow cell which was run on your sequencer. It's a very fine observation. So we do not go in depth into these things, because our per base sequence quality is good. Here also this shows exclamation, means it is not 100% like a ideal state, but it is a good quality data. So the next important check is per sequence quality score, means every sequence. See, we have how many we had several thousands of sequences. Here it is showing the number of sequences, and here the quality of the those sequences. So majority of the sequences you see which our first read one has captured is above Q30, which is was shown in our this figure also, but it is explained in terms of number of sequences. So majority of the sequences have score above Q30. So this is also a green tick here, because it shows that the good quality sequences are present in our for read one. Then if then the base content, that is ATGC, how much percentage of ATGC is present here. T is represented by red color, blue is representing your C, and A is representing your green, and G is representing the black. So see, you see that in the 150 base pair read from 1 to 150, you see that in the initial region, it is a very commonly observed feature that when the sequencing just start during initial 10 base pair priming of the calling of the reads of the bases, it generally doesn't give a very uniform result. That's why it is a haphazard. It sometimes gives only C, or it only gives T, due to which the initial read is not so good. So in this case, in such cases, we trim this part. In the next step, I will be telling you that we will trim it, remove it, so that these bases, the poor quality bases, get removed. Then after 10 base pair, you see that there is a uniformity of calling. It is not so that uh, one specific base is being called higher rate. Every base is in ratio of 25%. Okay. So this we will remove these reads, and because this initial part is not so good, that's why this per base sequence quality is marked as cross. Then comes the per base GC content. The per base GC content of the ideal state is like uh, the per base GC content is like a ideal curve. If it is ideal, it is around approximately around 50% in majority of the reads. Means this is the number of reads, and this is the GC content. So it is ideal. The state number of reads per GC content in the ideal state is this. This is a theoretical distribution. It is never observed to be theoretical. It is always a bit different, because theory, so the GC content theoretical is blue, and this red one is what is observed in our case. So majority of the reads, if you see, have a GC content around 50 only. But few reads are there which show a little divergence, but it's overall a around 50. That's why it shows a green color, that GC content is okay. Then comes the per base N content. N represent the N comes in a sequencing run read when that region is not called, that base pair is not getting called. So here you see that N is completely zero. There is no such N present, only here a little higher is there. It is negligible. So all the bases in the read present are being read by the sequencer, and we get its sequence. So it's very good for in our sample, and this is observed in the read one, and that's why it is green. then if we see the sequence distribution length, as we read in our basic stats, that it is 151 base pair. So we see that it's very ideal. The majority of the reads are of 151 base pair. This is the number of reads. This is the sequence length. So because majority of the reads are very are 151 base pair, so it's very good that we have got a uniform read length. It's not that we got shorter reads due to failure at a step in sequencing run. So this is a good data to be used further. Then comes the sequence duplication level. Basically, what happens that during sequencing run, when the PCR reaction takes place, there are sometimes very high duplication of sequences there, and there are deduplicated sequences here. It is there, but it is in the sequence duplication. If you see the level, and then you see that this is the total percent of sequence. This is the compared to the total sequence, the deduplicated sequence, which is red and marked by red line, is very less. So I feel that there is no issue in this. There is not a very high issue. It has been indicated cross here because it finds a very ideal state. That's why it is, because little bit there is sequence duplication in your means the total in the total sequence and the deduplicated sequences. There is a little bit hunch at places. That's why it gives you the red curve, the cross. Then there are some sequences which are like this regions. These are only found when your data, when your adapter, which I yesterday told you, get attached to your input sample DNA. That adapter gets read during the sequencing run, which should ideally not get read by the sequencer. Then it shows the overrepresented of these sequences. So this should be if this should not be present. If it is present, then we will remove this during, because it is present, so we will remove it before, in the trimming step, before going to the further steps. So you see, but if you want to hear, this software also does, it counts the number of such overrepresented sequences. It also counts the percentage, and it also gives the possible source if it can. But here it didn't find any possible source, so it doesn't give. And it also separately gives the what adapter contamination. The adapter contamination is clearly present in all these reads, because after 60, if the position of read is 1 to 150, after 60 base pair, there's a continuous presence of this red color is Illumina universal adapter, because Illumina was used for sequencing this sample. It was obvious that the adapter sequence of Illumina must be present here, and it was means it. If there is an adapter, then it must be of Illumina. Otherwise, ideal state is that there should not be any adapter contamination, means there should not be any adapter region read by the sequencer, the sequence of adapter, because adapter were additional regions to for the binding of your input to the flow cell. But here, because adapter is read, that means your input fragment size was smaller at some place, got due to during the step of fragmentation, or during this step, all the libraries preparation step. That's why we have to remove these adapter sequences, other we will misinterpret our data. So as I explained you, that this is for the till here, did you understand, so that I can explain you the read too? Okay, someone asked me that what the whiskers say, ma'am. What whiskers, which whisker you are talking about? What color mean for each line? Yeah, what is not clear? Can you speak out?
Hello, ma'am.
Yeah, please say.
Yeah, in the after basic statistics, per base quality sequence, the second one.
Yeah.
Yeah, in this one, the box plot is showing the whiskers, right? Below this one. So what these whiskers say about, ma'am? Like what they
these you
indicate?
By whiskers, you are meaning these lines which are the
Yeah,
range,
yeah, yes,
range.
ma'am.
Yeah.
Yeah,
Actually,
yeah,
actually what is there, that this is the 150 base pair read. Okay. And there are thousands, we have thousands of read. So because every read doesn't have an exact quality of Q30 of Q30, like it is showing here, it is this region, this is a range. Few reads are in the range of Q36, few reads are in the range of Q32. So this line gives an around average median region of that, of your number of your quality of your reads at this position. So this is the range
yeah.
of all the
Okay.
reads at this position, and this is the middle point, is shows the is the median, or you can say an average, which reflects the average of the total reads. Did you
Yeah.
get my point?
Yeah, yes, ma'am. Thank you, ma'am.
Okay. Any other query? Then please interrupt me at this moment.
So if you have any query, you can unmute yourself and ask
Yes,
it directly to ma'am. Otherwise, we will proceed.
yeah, Isaac, yeah. Please, Prof, may you come to this sequence duplication levels? I didn't understand some of those color codes lines and how actually the duplication, where is the duplication or where there is no duplication. I didn't understand.
Which color line? The sequence duplication level you are talking about?
This sequence duplication levels
Okay, okay.
on the
I'll
left side with X.
yeah, this. See, there are two lines. It is present mentioned here that the percentage of sequence re remaining if deduplicate is here, for overall percentage is 43%. Okay. And how it is calculated? The deduplication, that the total sequence, what was the total sequence? This blue line shows this was the total sequence. Based on the sequence duplication level, it is plotting from 1 to how much is the duplication level. So if it plot from 1, majority of the sequence are duplicated only once. So this blue line is at high here, and the red region is shows the deduplicated sequence. The blue line is the actual total, and those sequences which had duplicated. So majority are very less duplicated because it is at the one duplication level, but few are there which have also have a 10x, above 10, above 9, above 10 duplication level. So these reads will be removed during the trimming step.
Mhm. Okay, thanks.
Is it clear? Okay.
It's clear now.
Any other query? Okay, then I'll proceed further. So for similarly, the stats will be calculated for the all the quality parameters for the read 2. If you see, the basic stats for read 2 is also very similar to the read 1. The per base sequence, but if you see the per base sequence quality of read 2 is much poor compared to read 1. This shows that during the sequencing run, read 2 was still better captured compared to the read 1. So you see how poor it is. So we will trim it very properly. You see that the initial basis from 1, 2, 3, it the dip comes below Q Fred Q score 20 also. There is so poor basis being called. So, but the region after 10 base pair, if you see, is of good quality, and it goes approximately, it is above Q30, it goes up to 140. So this picture suggests us that we should trim the reads from the forward and the reverse region of the our reads of our read 2. And as we have seen that in read 1, the initial part was not good, the initial 10 base pairs, we will be trimming that also, so that we do not get this low quality reads in our further run. The per tile sequence quality is not a very it was not good there also, because because initial, this shows the initial region on the tile on your flow cell, this initial region was not captured good. that is clearly shown by your sequence quality plot. That's why it is not showing the bluish region. It is showing reddish, yellowish in the front part and also in the back region. Then comes the per sequence quality score. As seen that majority of the sequence have a good Phred score, but lesser compared to the our that data, our read one data. If you see read one was better compared to read two. See above 35, 3,50,000 reads were good quality, but here if you see it is not so. Only if you see it is around 20, 10,000, around 2 lakh only reads are above good quality Phred score 30. So here lesser reads have a good quality score. If we see the per base content, sequence content, then we find that here also the initial part till 5 base pairs have lower quality compared to the rest. It means the lower quality in terms of calling of ATGC. It is not uniform 25% of ATGC. It's similar, it is very similar to the our read one, but read one here was still 9 base pair. Here it is poor only till 4-5 base pairs. Then comes the GC content. The ideal GC content is almost a bell-shaped curve, but our is also nearby it, as I have explained in read one. That's why it shows this type of curve. Then if we see the n base content, it is very negligible. It is very good in both read one and read two. Sequence the length distribution is also very good in both read one and read two. If you see, all majority of the reads are at 151 base pairs, which is very good. And then sequence duplication level is not very ideal in here, just like read one. It is very similar to that read one. Overrepresented sequences are a lot in this data, so we have to do a very good trimming in this data set. This is the set of overrepresented sequence. And if we come to the adapter content, it is again the adapter contamination after 60 base pair, and that is present in the Illumina universal adapter contamination is present here, which we have seen earlier also in the read one. So I feel this part I will now come to the next step. You all got this result when you have run fast QC.
Actually, I'm using a Linux platform. So, and the files are in the form of RAR folder. So I have to install one software to unpack. For that, I'm doing it. I will do it.
Yeah,
It will take some little time.
yeah, you can use. You can
Yeah, I have
do
installed it.
using
So yeah, and RAR command I have to use.
so I should wait for you or I should proceed?
You can move it fast QC. I will do it. In maybe 7 minutes it will be done. So you can
Okay.
move further, no problem.
Okay. So the next step, we have discussed all about quality check, okay, of all these reads, which I have shown you for one sample. You have to run for all the samples which I have given, and that's almost similar because I have done all those samples. I found the similar results. So I saw that how does the quality reads obtained from sequencing data can be analyzed by this fast QC and also with NGS QC and FastP. And there are many tools nowadays, multi QC and those things. Once we see the quality of our reads, the next step is we have to remove the low quality reads, and the adapter can be removed using Trimomatic software. So next our run will be Trimomatic. This here in the slides I have actually explained you all those things which I have directly explained you on the output of fast QC. So there I was explaining you the quality. I can explain you exactly what is the Phred or Q score, which we call. Q score are defined as a property that is logarithmically related to the base calling error probability. That is that what is the probability? If Phred score is 10, then it is a probability that one incorrect base can be called in 10% in 10 base calls. That is the accuracy of the call is 90%. If Fred score is 20, then there is accuracy of calling one wrong base in 100 base pairs. So it's 99%. If Q score is 30, then the accuracy of calling one wrong base pair is 1000, so 99.9. So here it shows how Fred score is important in a sequencing data. And here there is a comparison of two platforms of Illumina, MySeq and HiSeq. How much percent of good quality score above Q30 they give, how much bases they give. This is a very platform specific result, this means table, so no need to go in depth of it. So next, all these I have explained you, all these on the output only, so I will not repeat it. So now comes the Trimomatic. I have given you the link for the Trimomatic. Trimomatic is a flexible read trimming tool for Illumina NGS data. This software is used to remove adapter and low quality reads and bases for raw reads, which are identified in the quality checking steps. So now, first of all, you have to download this software. I have this software, so I can show you how it runs. So once you will install the software, I've given you the link. The link is very simple. It's very easy to install it. You just have to take these binary files, the source files, and then you have to install it. Okay, it is a zip format and it will give you it will run. So once it is installed, should I wait for your installation or I should proceed? Please, if someone from Decode is there, can suggest me.
So you can continue. I will share the video recording of this session as soon as possible, probably 1 or
Okay.
2 hours.
Okay, sure.
But
So,
please share the installation link in the chat box or
yeah,
somewhere.
I have given the installation link earlier only in the chat box. I will share again if you say. This is the link. This is Trimomatic installation. Okay. So now what we will be doing for the Trimomatic run, we have the Trimomatic commands needs the input of whether your read is paired end. What is, you have to mention it. What are the number of threads you want to use? What is the Fred score quality we want your reads to be trimmed till? You have to give you can get the summary stats file. You will give input the first read, the second read, what will be the output base you want in your files, and it also takes the information of the adapters, which I will explain you in my command. You are able to see my slide, right? I will check once.
Your slides are visible.
Yeah. So see, the command consists of this Trimomatic paired in the threads I've mentioned is 20 because I have a very robust lab system. So you might, if you don't have these many threads, then don't use the thread 20. And then you will give the input of the sample 1, that is the raw read, which we have discussed, whose quality we have checked, sample 2. And you will also give what output file you want to name for the forward read, for the paired forward read, because it will trim and give you 4 files. One is read 1 forward paired, other is sample 1 read forward unpaired, the other will be sample 1 output reverse read, that is the R2 read, which is paired, and also the sample 1 output reverse read, which is unpaired. Along with it, because we have to do the trimming here, so we will give it as an input, which I have sent you in your in the RAR file, which today I have sent in that there is a file present Illumina universal adapter, because we saw that Illumina universal adapter contamination is present. So this file is given as a input to Tremomatic that remove all these. So Illumina clip function, what does it do? It cuts the adapter and other Illumina specific sequences from the read. And what does this and these different parameters are basically indicating that how much stretch of the adapter is to be removed and in what region. Okay, we have 10 upstream and downstream to be cut from 10 upstream. The 10 around 10 base pairs of the alumni adapter contamination was to be cut, so we have mentioned it here. And because the leading 20 base pairs, the initial part was not good in our reads, so we have mentioned that leading 20 you have to cut. Trailing back end 20 also please trim it. That is cut the basis of leading parameters indicates that cut basis of the start of a read if below a threshold quality, and trailing is cut the basis of the end of the read if it is below a threshold quality. Threshold is here our Q30, that if it is below Q30 then cut that region in the trailing in the back end of your read. And min drop length, that is if any sequence has a drop length below the specified length. Here we have specified that minimum length should be 36. If the read has length below 36, then you drop it. So min length, drop the read if it is below the specified length. Specified length is 36 base pair here. So these all parameters we will be using during our run. So I'll copy this in your chat box also.
Excuse me, ma'am.
Yeah.
I have a small doubt, ma'am. Now we are having only 2 samples. If you are having
Yeah, right.
multiple samples, like 50 or 100, then how can we use this command, ma'am?
You have to write a script which you have to put a loop which will read each files one by one, or if you want to run it parallelly, then you have to give a parallel command.
Okay.
It's
And
it is,
whatever,
yeah, you have to write a script with a
yeah,
loop. Yeah.
and whatever we have mentioned at the last command, like adapter true leading is equal to 20, trailing is equal to 20,
Yeah.
and minimum length is equal to 36. These are the standard readings. Like we have to change according to the fast QC report, ma'am.
Yeah, it should be changed according to the fast QC report,
Oh, oh, okay,
because
ma'am.
that is the reason why we did fast QC, right?
Yeah, yes, ma'am.
Yeah, that is the reason we did fast QC. And if, but general it is by default. Also Tremomatic suggests some parameter, but if you keep in mind your fast QC result, then it would be great. Then you will not be losing your data. If the quality of my reads were good in the forward or reverse region, I won't have used this leading and trailing parameter. And if I didn't have any Illumina adapter contamination in with my fast QC, I won't have used this.
Oh, okay, ma'am.
Got it?
Yeah,
So, okay.
ma'am.
So now
Like
I'll
how
come.
to how to write the loop, ma'am?
That loop is is like coding. How you write a for loop.
Actually, I am new to this one, so that's why I'm asking, ma'am.
So
Like,
that that can be a separate session for coding, how to write codes for running for loops and all that is a coding part, right? That
yeah,
how
yes,
you
ma'am.
code. It is not. This is not the part of running this tool.
Oh, okay.
So here I have copied that command, and because I have all the samples present here, so it will run. See. It is so clearly saying that these are the Illumina clips which were present in your data. And it will take a bit time. It shows that the quality encoding detector is FRET 33. It will take, after some time, it will get completed. because the size of the read. Similar thing we have to repeat for all our input sample. See, for any GWAS analysis, the sample size, the size of the input sample is always kept to a higher end. It's above 100 generally has samples, because then only statistically we get a significant result. So here I just have shown you for the one sample with a paired and read 1 and read 2, and the data set which I have shared with you that contains more than one sample for which I have done the analysis. But here at in the time constraint, only one sample can be run, because I am running on such a high thread 20, so it is taking time. So if you have lower threads, then it will take more time.
Can I ask you one question, please?
Yeah, please, you're welcome.
What is this data set actually about?
What is this data set?
Yes, samples.
These samples are the output of GBS run. As I
No, what
explained,
is the organism?
organism,
Yeah.
the data set organism is a, because it is a read generated from a animal sample.
Okay.
So after the GBS run,
Okay.
genotyping by sequencing,
This is from animal.
right? See, now the output came that the input reads which we gave was this much, 68,000, 6,87,599, and the reads which survived both, means both forward and reverse, were around 99%, and only forward surviving were 0%, means majority of the forward had their paired, and reverse surviving are only reverse are 209, which is 0.03%, and dropped reads are 128, and the trigonometric run is complete. So now we got the set of 4 files, one with the paired and read of the forward, one with the pair and read of the reverse, and the 2 files with the unpaired reads. Have you run till here? Then I will go to the next step. In the next step, we have to install the software BWA, which is a aligner. BW is a very popular Burrow-Wheeler aligner, which we will be using here for aligning our sequences to our reference genome. So I will give you again the link of the software. I have earlier mentioned it, but I will give you again. It is a Burrow-Wheeler aligner, which is Burrow-Wheeler. BWA is a software package for mapping low divergent sequence against large reference genome. Okay, I'm sharing you the link again. You can download it from here, or if you are using Ubuntu, then you can type this command sudo apt install BWA. It will itself install this software. Are you getting my point? Hello?
Yes, ma'am.
Yeah.
Yes, ma'am.
So the next step now we will be doing is the BWA. We will be using BWA for the alignment of our reads to the reference. So for running BWA, first we need a reference genome. You understand what is reference genome? Can anyone answer me what is reference genome?
A genome of that organism which is already annotated is called as its reference genome.
Exactly. That is the reference genome, because here we started saying that, now you must be able to see my slides, that we have to we are identifying variants using a reference genome. So here we are now, before alignment, we have to index the reference genome, and here we are using BWA for indexing and alignment. You can also use HiSet build or Botai2 for this, but here I will be showing you BWA. So BWA, you will type BWA, then you will type index, and then you will type the sequence of the reference genome. This will generate the reference genome, and this will be the, I will share the command with you. I have shared the reference genome sequence to you. You can use that sequence. It is named by reference genome underscore genome.fa. It is a little bigger size, so it will run like I have run BWA index reference genome. It will take some time. After completing the iterations, it will give you final the several indexes. In the index files also I have mailed you. I will show you here how you will run.
Hello. Hello. Excuse me, ma'am.
Hanji.
Ma'am, if you are having, if you want another reference genome, like we are testing another any other organism other than this, where from where we can download this reference genome, ma'am?
See, whenever a reference genome is sequenced, any of the reference genome who against whom you want to check your data set, the reference genome of every published organism is on NCBI. You can get the check the reference paper, and you will get the NCBI submission ID, and from that ID you can go to NCBI and download it.
Oh, okay, ma'am.
Okay.
Like we have to download in faster format, ma'am.
Yeah, it is available there in faster format, and you have to download in faster format.
Yeah, okay.
Okay, so I feel you must have installed BWA using sudo
Yes.
apt install BWA. Now I will be running BWA. See, BWA index is used for indexing the genome. So as I have already shown you that how it was running, we will give the BWA index and the reference genome file. .fna file is there in your, so the command will be BWA index reference. It will generate, it will take a time, means around 20 minutes it will take. So I will not run this command currently. So this reference genome run will, what it will give as output, I will show you what files will be generated, because if I start the run, then the output file which I've already generated will be rewritten. So I will show you what output files I've generated. See, from this, after running of that command, these many output files will come. The different, because it is indexing the genome, and after indexing, it gives the .amb, .ann, .bwt, .fai, .pac, and .sa files. These all files are the output when you are using indexing using BWA index. And if you are using some other software, you will not get this file. It like Botai gives .bt2 file, .ref file. So it depends on how the algorithm is based of that software. So now we will be using these files for our alignment step. Okay. And for before alignment, I want to ask you that do you understand what is alignment? I should explain you. I feel I will explain you.
Excuse me, ma.
Yeah.
Please, ma, this reference and the genomes we are using, is it the organism, is it human being?
Currently, the organism which you are using is not human being, which I have given, shared you the slide.
Okay, the other question I have, ma, is it whole genome sequence or targeted?
It is whole genome sequence.
Okay, thank you, ma.
Yeah, it's a good question. It is all, you should always take a whole genome. The higher the quality of your genome, the better will be your result. Because sometimes some organisms have draft genome, and those draft genome, unfinished genome, always have some errors in them. And as the quality of the genome gets improved, we get a better result. So because everything is based, whatever you are, if it is reference based alignment, then all your final output are based on your reference, the quality of your reference. Okay, so now the next step after, once the indexing is complete, means this step is complete, this run gets complete. Anyone has run BWA index has started running? Is it running?
Yes, ma'am, it's running. Bharti
Yeah,
ma'am is
okay.
saying running. S. Kuvar is also saying running.
Okay, great. Then I will explain the next step, the mapping step. In the mapping step, now we have to map the FASTQ files, that is the obtained from the Trimomatic output, to the reference genome. That can be done using BWA, HiSet or Batai. Here I have used BWA. And what is mapping? If anyone wants to understand that, it is, we are aligning our raw reads which we have obtained against a reference. So the alignment, it is aligning to a reference. If you see, this is our reference, these reads which we are there, they get aligned, means the complementary match where they get aligned according to the match, and they get stitched together so that we can actually get the alignment file, the alignment result specific to your reference. So what out command we will use here is BWA mem. Mem is a function of BWA in which is used for alignment. There are other function also in BWA. I will explain you. This is a good thing to explain. See, BWA is a software for mapping low divergence sequence against a large reference genome such as human genome. It consists of 3 algorithms: BWA backtrack, BWA SW, BWA mem. The first algorithm is designed for Illumina sequence reads up to 100 base pair, while the rest two for the longer reads, means BWA. It is saying that the first algorithm, that is BWA backtrack, is designed for Illumina sequence up to only 100 base pair reads. Means if the read is more than 100 base pair, it is not used. While the rest, the BWA mem and BWA SW function is used for the reads which are higher than 100 base pairs, whose size is higher than 100 base pairs. So, and it can be range starting from the longer sequence can be of range 70 base pair to 1 MB base pair. So BWMM and BWSW have share very similar feature, such as long read support and split alignment is also there. But BWMM, which is the latest and generally recommended, that's why we are using BWMM, is for high quality queries, is faster and more accurate. BWMM also has better performance than BWA backtrack for 70 to 100 base pair Illumina. So BWMM is the most ideal. So we are using that BWMM here. So I'll be showing you how to run this BWMM. So you have to run this command BWMM. I will copy this command and give you in your chat box. You will run BWMM, the threads, how much you want to give, and then you will give nice. Then you will give the FASTQ files, which you have the FASTQ files sample1, fastq.gz, and sample1 forward. Means we have got 4 files in that after Trimomatic run, but we will be using only the paired files. We will discard the unpaired reads. Sample1 forward paired, sample1 output reverse paired, and we will get the alignment file. And this will be the run. I will share the command, and the run will go as shown in the screenshot below. I don't know where is the chat. So you all can, and I have named it as sample1 underscore SAM because the output file is in the format of SAM. The output file generated is SAM SAM is an alignment format. It is a SAM file, and we have given GZ so that it is compressed. It doesn't take a lot of space. Okay, I hope you understand this, and you can start running. I will also run here. I have given you all the raw files, so you can run it. I will give the path where I want to save my file. Okay, see what I've given here: BWMM, the threads, the reference genome, the sample one. The I have given 40 threads. You don't give 40 threads if you don't have 40 threads. Please be cautious, otherwise system will hang. Then you can remove this option also if you do not have many threads in your processor. Then sample one forward and sample one reverse read and the output file and it SAM file and its path. So fail to locate the index sequences, it is saying, because indexing was not complete. First, I should complete my indexing. BWMM index. If your indexing is complete, then it will not show that error. Indexing will take a little bit time of the genome. Once this is done, this command will run. So let it run. I will explain you a little bit theory left for this today's session. I want to explain you what is SAM format actually. SAM format is sequence alignment format. You can see my slide, I feel. So SAM format, and what is there in the output SAM file which will be generated after the run which we have run. We will get the read name of the read. We will get the flag. The position of flag is actually used to highlight some points. The chromosomal region, the position of the chromosome in the chromosome, at which point it is, what is the mapping quality of that alignment. Then comes the cigar string, which represents the quality of the reads. The mate position means the when we do paired and run, we give the mate position. If we have a mate sequencing, we have used mate pairs, then it gives the mate positions of the chromosome. The read key what is the sequence of the read, what is the quality of the read, and these things contain the edit distance at the end. It is the match string, what is the region is matched, what is the best alignment score, the next best alignment score, and what are the read groups. This is the total information in a SAM file. So I feel you understood till here. Reference genome is still running, indexing is still going on. Once it gets indexed, this command BWMM, you all also after indexing run this command so that your alignment file will be ready today. So by the end of this, after 1 hour, if you have lesser threads and all, your 1 or 2 whatever time it takes, be ready with this SAM file so that we can proceed for the further all these steps tomorrow in our class, and we can wind up the session of reference based variant calling, because now it will take time, especially who have a lower configuration of system. So if you all are okay, then I can stop this session and you all can also complete at your end.
Hello, ma'am.
Yeah.
I would like to request you if you could share a few basic paper of Jivas, so before we can read and we can understand. It will be easy for us, I think.
Yeah, I will share the papers.
Okay, thank you.
But the thing is that you want general papers or a bit means you do not know at all about Jivas, or you know about
No,
it.
I know, but I would like to because I am not expert in Jivas, but I need. I mean, I would like to understand this session properly. So
Okay.
in that scenario.
Okay.
You
Okay,
can send me you can send the both kind of paper, ma'am, like basic people's like advanced kind of. So because I don't know anything about that also.
Okay, okay, sure. Anyone else has any
Thank you, ma'am.
query? Then
Ma'am, there is one query in the chat box.
Okay.
So Padmani ma'am is saying, what is the meaning of low divergent samples?
I'm not able to get that question. What is the meaning of low divergence? Low divergence means the diversity is low. By diversity, you means that the samples are not different, that different. And how the difference is being measured here, it is based on the variant, that the SNP, the variants found in the samples are similar, not so different. So if it is low divergent, diversity is less.
So
At a time, you can use a single reference genome, but if not together two, but separately you can run two reference and get the two SAM files.
Ma'am, one more question is from Ravi Kiran, who is saying how to do de novo indexing if you don't have reference genome.
That will be your third class in the later classes, because de novo is when we do de novo from these reads, which the input raw reads, we built a reference. So that I will explain you when we will be doing the de novo session. This session was reference based variant calling.
So thanks, ma'am, for
Okay.
it was a very beautiful presentation, and uh, you answered the questions of the participant beautifully. We are thankful that you delivered on our platform for making this workshop wonderful. So I will share the material with our participant as soon as possible, like video recording and this chat.
Yeah, sure.
So we will meet again tomorrow at the same time, that is 6 p.m. Indian Standard Time, with third session on genome-wide association studies. We believe you are enjoy learning with the code. Like, thanks. And now I seek permission of ma'am for ending today's session.
Yeah, you can end, but I can request all participants run for all the samples I've given you, sent by the mail, run the alignment for all the samples. Okay.
So thanks, ma'am. Now I am going to end today's session.
Okay.